Hello, everyone. So, as part of this presentation, I thought it would be nice to just get a quick visual representation of what the complement pathway actually is. Now, in Guillain-Barré, normally there's three different types of classical pathways. There's the classical pathway, the lectin pathway, and the alternative pathway. They all do different things, as we can see, but the one that we're focused on is the classical pathway. Now, it begins with the antibody binding to the surface antigen on the microbe. Now, once the antigen binds, it causes this protein that's just floating around in our blood, always just there, hanging out. It activates it, right? So that's it. In the classical pathway, it's a complex. It's a C1q, they call it. C1q is made up of C1r and C1s. Now, when the antigen binds to the antibody, the C1 complex becomes activated. When activated, it causes the hydrolysis, cleavage, of C4 and C2. Now, C4 and C2 get broken up into C4a, C4b, C2a, C2b. C4b and 2a then form a complex on the pathogen surface. And this complex is what we call C3 convertase. C3 convertase converts C3 into C3a, C3b. Now, when C3 is converted into C3a, C3b, the C3b portion then joins that complex of C4b and C2a, and it creates this new complex that's created is called C5 convertase. Now, C5 convertase converts C5 to C5a, C5b. C5b is then able to bind with some other complement proteins, C6 and C7, and when it joins with C7, it causes C7 to change its conformation and allows it to insert itself into the pathogen surface, the pathogen cell membrane. When that happens, it allows C8 to bind with C7 in the cell membrane, and C8 allows C9 to bind with it. So it's this whole complex, right? So C5b, C6, C7, C8. C9 is a cool one, though. C9 has two different sides. It's got a hydrophobic and a hydrophilic side. The hydrophobic side is what binds with C8. The hydrophilic side is what makes the pore. We'll talk about that in a second. So C9 agglomerizes, right? So what that means is it basically makes copies of itself. So it inserts itself in the cell membrane, makes a whole bunch of copies of itself, and creates this pore-like structure. Now, the outside of the C9 pore structure is hydrophobic. So it allows it to be inserted into the lipid bilayer of a cell membrane. The inside of the pore is hydrophilic. Now, that means water-loving. It's pretty much the opposite of a lipid. And this allows this pore formation to form. And what it really does is it allows the water molecules that are outside of the cell to enter the cell. Water molecules sodium ions, different ions, and water molecules are able to rush into the cell. Now, when I think of it as like a water balloon, when water rushes in to a water balloon too fast, it inflates and then pop, right? And that's exactly what happens here. And we call this lysis. It lyses the cell and basically destroys it. And that's what happens in Guillain-Barré syndrome, where the classical pathway gets activated, the membrane attack complex, which is that pore, gets formed, and the cells get destroyed because of these water and different ions that are able to free flow into the cell. And now, I just thought it would be nice to have this visual representation of really what's going on, because I feel like it gets a little confusing with the 1s, the 2s, the 3s, the A's, the B's, but when you really think about it, C1 activates C2 and C4. What are we missing? C3. Oh wait, there's a C5. Oh wait, C5 gets 6, 7, and 8. Oh, what happened to 9, 7, 8, 9? Well, 9's redeeming itself, pretty much. 6 is no longer afraid of 7 because 7, 8, 9. 9 said, I'm not going to be eaten. I'm going to redeem myself and I'm going to kill these cells.